Namaskar. Good evening to all. On behalf of Indian Society for Veterinary Medicine, I welcome you all for today's webinar. I do request Dr. Ramprabhu sir, General Secretary, Indian Society for Veterinary Medicine, to brief about the webinar. Ramprabhu sir, please.
Thank you, Dr. Galdhar. So, it's my privilege to welcome Dr. Sandeep Bhatia, Principal Scientist, ICAR-National Institute of High Security Animal Diseases, Bhopal, for today's webinar on pandemic preparedness, role of veterinarians. This topic chosen by Sandeep Bhatia is of importance to all the practicing veterinarians as well as the faculty members and students of veterinary colleges. So, on behalf of ISVM, I welcome you, sir, for the webinar. I also welcome all the participants and life members of ISVM, PG students, and other veterinarians for this today's webinar. I welcome one and all. Thank you.
Thank you, sir. Before start our webinar, I would like to introduce about today's speaker. Today for ISBM webinar, we got a speaker, Dr. Sandeep Bhatia sir. Just in brief, I would like to introduce about Bhatia sir. Dr. Sandeep Bhatia obtained his BVSc and AH in 1991 from GB Pant University of Agriculture and Technology, Pantnagar, MSc in Veterinary Technology in 1993 from Indian Veterinary Research Institute, and PhD degree in Veterinary Microbiology and Immunology in year 2005 from Mathura Veterinary College. Dr. Bhatia joined ARS as scientist in 1997 and has been working since 1999 at the prestigious High Security Animal Disease Laboratory of IVRI at Bhopal, which is currently an independent institute known as National Institute of High Security Animal Diseases and is presently holding the position of principal scientist. During his 20 years of service at both the lab, he has made significant contribution to the development of diagnostics and vaccines for emerging viral diseases of animals, diseases like bovine immunodeficiency virus, bovine viral diarrhea virus, and avian influenza. He was awarded National Fellow Project in April 2011, in which he pioneered the use of pure genetic for avian influenza in India for developing reassort avian influenza virus as the biomarker vaccine candidate. Currently, he is working on concept of universal influenza vaccine using recombinant DNA technology. His other notable contributions are in diagnosis and surveillance of H5N1 virus in poultry, bovine immunodeficiency virus, bovine viral diarrhea, and malignant catarrhal fever in cattle and buffaloes. Dr. Bhatia was also awarded with USDA-sponsored Norman Blocker Fellowship in 2008 and was conferred all-round excellence around award for best performance in summer school on team building at NAR, Hyderabad in year 2006. His other achievements include development of a disease data management system for avian influenza in year 2009 and import risk analysis for avian influenza for livestock and poultry products. Dr. Bhatia is one of the few assessors of National Accreditation Board for Testing and Calibration Laboratories (NABL) for ISO 17025 in the field of veterinary diagnostics. I request Dr. Bhatia sir and welcome him to deliver today's webinar. Dr. Bhatia sir, please.
Thank you, Dr. Galdhar. Thank you so much, and I must first thank the organizers and the ISVM as a whole for providing me this platform for sharing my experiences. And I chose these topics because we have been involved in last 4 months for COVID diagnosis, and we were aggressively working towards it. And before that, actually, as the topic is pandemic preparedness, at the National Institute of High Security Animal Diseases, our main mandate is preparedness only. So our mandate is for exotic and emerging diseases. So since last 20 years, we have seen how preparedness for an unprecedented disease can be planned, and what we have to do as a whole. But now this time after COVID, actually COVID-19, we have experienced that we have so much to share and contribute to a human disease also. So as a veterinarian, we feel that we have lots to contribute for zoonotic diseases as a whole, and for pandemic prevention of pandemic also, veterinarians play a very critical role. So I thought we should have a discussion on that, and I will initiate the discussion with my few slides, and then we can discuss on that. And all the audience, actually, I request to put their provide their inputs wherever possible. And because as a field veterinarian, you have a different kind of role. We have been working, especially I have been working as a laboratory scientist. So we have a different role to play. As a field veterinarian, you have a clinician or a veterinarian working in the clinics. They have a different role and responsibilities. So we have a diverse kind of roles as veterinarian, and it is very important. As this One Health concept, it is being talked about. We know that everything is now connected. It is realized now. It was connected earlier also, but it is now so much realized after COVID-19 that human disease, animal disease, veterinarian, medicos, the environment, everything has to be seen as one concept, and then we have to address the problem seeing under the One Health concept, so that we can comprehensively and then very rightly actually address these problems and then bring out the solutions. So if everything is set, should I start the my slides? Share my slides with the...
Sir, please share your slides.
Yeah. I hope you are all able to see the slideshow.
Yes, sir.
Yes, sir. Yeah. So before we talk about the present and future, we must just have a quick look on the past of the pandemics and epidemics. As we know, it has started since long. Every 200 years or 100 years or maybe 300 years gap, we have one kind of pandemic. But the earlier ones were very much different than the current ones, and the future ones may be altogether different. So just have a look. The history says that Anthony Plague in AD 165 and to 180, it was a smallpox or measles in Rome. So it started with this. This is the earliest record of a pandemic in this globe. And then after that, we have the plague of Justinian. That plague is means Yersinia pestis, again bacteria in Europe, and then followed by the Black Death again. This was a big pandemic, and it was for 8 years. 7 to 8 years it remained there, and it was again Yersinia pestis. And because at that time actually antibiotics were not so much developed and discovered, so most of the epidemics and pandemics were the causative agents were actually bacteria. And after that, we had this American plague. It actually, this Yersinia pestis as plague, it continued in every century from 1300 to then 16th century. Number of epidemics and pandemics, they took place actually. Then Great Plague of London in 1665 and 66. This is recorded. And then came the flu pandemic. Possibly at that time, actually, virology was also not so much developed. and they think that possibly it was H2N2 influenza virus, but there is no proof actually for this flu pandemic of 1889 and 1890. Then came about which we all know the Spanish flu, the 1918 to 1920. That was H1N1 virus. About this virus we already know, and they have reconstructed of this virus also to study how the future pandemic can be. And after this pandemic, actually the biggest pandemic has happened now on COVID-19. Of course, by definition, actually, a number of other pandemics have happened, like the H1N1 swine flu. That was also pandemic H1N1 virus, we call it, in 2009. So, but the size and the numbers, they are very much different right now. The current pandemic of COVID-19 is actually of the size, much lesser size than the Spanish flu, actually. So millions of people, they died. The death rate was very high because of the medical facilities and other things. So of course, there are certain other factors also which were not present, which led to this fast spread of COVID-19. The globalization was not there. Intercontinental trade was also not there. So there were no long route journeys were not there. Air flight journeys were not there. So with that, actually, the spread is actually now the very fast, and then we have to prepare. The only way to actually control pandemics and is by preparedness. So earlier we used to hear and read about the pandemics, and then the most, as per the risk assessment, actually, the most risky virus was supposed to be influenza, because the last pandemic was from the type A influenza and H1N1 virus. So because of the easy spread and the nature and because it is a RNA virus and it can mutate to, and there are so many subtypes, and it can enter into variety of hosts also. That is also one of the factors. So we heard about it, and then we had some kind of preparedness that I will tell how we prepared for flu pandemic. Actually, we kept on, and then let us have a look of the current scenario. So this data is from the OIE website, the current data. The risk in the zoonotic infection is increasing actually, as animal pathogens, more and more animal pathogens, they are adopting the human beings. They are crossing the species barrier, and with a greater ease. And there is climate change also coming into play and facilitating the spread of the virus from wildlife to domesticated animals, and from domesticated animals to human beings also. So as the population explosion is there is a rise in the demand of food. So for more food, we have to produce more and more number of animals, like in poultry, as we have seen in case of avian influenza also. Avian influenza, the rise in avian influenza cases and the epidemics, it rose as the poultry grew. So when you have a substrate population, the susceptible population growing in numbers, then virus has more chance to grow actually. And then when there is a close contact between the animals and the human beings, then also the chances of jumping from animals to human beings, there is more chance actually. And for COVID-19, the current outbreak of COVID-19 also, this is one of the, suspicion and theory that possibly it jumped how it could cross the species barrier was because of the wildlife market, the meat market in China, that there was not much hygiene, there was not so much of separation, and there is a close contact between the wild animals and this human beings. So slowly and slowly, these viruses, they actually adopt, and then because everything, as you know, the mutation is always happening in RNA viruses. So whenever they get chance, some mutation will happen, and then they will be able to acquire a new property. But a sizable number of viruses can only jump into the human to next population or the different species only if there is a repeated chance you are giving it to the virus, so that 1 in 100 times it will succeed to cross the barrier. So that is the case. So as we see, the current scenario says that 60% of the human pathogens are zoonotic right now. And among the emerging diseases, 75% of the diseases are zoonotic as per the OIE. And the most of the feared pandemics, actually, the fear of pandemic will come from, it comes from the zoonotic viruses only, because they will cross the barrier, and then because we are giving a number of chance to these viruses to cross the species barrier. Now, the most, if we see how to control it, this kind of control, when it happens, actually, when it crosses the barrier, and then the kind of lockdown we saw in COVID-19, and the closing of the markets, closing of this, all the travel is closed, and all these things are actually not preventive measures. They were actually after something has happened. Before that actually comes the role of veterinarians. That is why role of veterinarian in preventing the pandemic is much more than the medical people or other policy makers or anybody else than a veterinarian, because they have to control at the source. So source is at the animal side. So if we take care of our animals, and then we do surveillance, then possibly we can reduce reduce the chances of emerging new virus, or giving them viruses a chance to cross the species barrier. So that is why the early detection of diseases and infections at animal source can actually prevent the transmission to humans, and they will not get introduced into the food chain. And that is why veterinarians, they play a key role in protecting health and welfare of the animals and protecting the health of humans also. So this is the central concept about it, that one thing is that most of the zoonotic diseases, most of the emerging diseases are zoonotic, and these zoonotic diseases or zoonotic viruses, they are going to become future pandemic. So we have to control these diseases at the animal source only. Then only we can prevent pandemics in an effective manner. Otherwise, after it has entered into the human population, then the cost of further preventing the spread is huge, as we have seen. Still, so millions and millions, they are going into this every day. Actually, within MP, we know that within Madhya Pradesh, actually, more than 1 crore rupees is spent only on the diagnostic kit for testing. So where it is going to end, we don't know. So beforehand, if we can do something, we can prevent so much of loss of life, as well as economic losses also, huge losses actually. So with this, with this SARS-CoV-2, we have learned a lot, and as we have been hearing about pandemic, reading about pandemic, but the real feel of pandemic, this virus has given us actually. How actually it happens, what are the issues, how to handle it, and this is a kind of so many things we have developed actually, and so many protocols, they are now set in each hospital, in veterinary hospitals also, in veterinary diagnostics lab, and the human side also. So many things have been developed, so that in bureaucracy also, in the departments, in the polity also, I mean, people now know that how to handle, right from IS people, the purchase people, the industry itself, they know how to deal with this problem of pandemic. It has also exposed the weaknesses of our healthcare system, and once it is exposed, we can do some corrections also. And the typical part of this pandemic of this size is that everybody is involved. It is not, you can't say that it is a doctor's problem, it's a human doctor or hospitals, they will handle it, they have to handle it. It is all people are involved. Veterinarians are involved, paravets are involved, policymakers, bureaucrats, politically, social workers also, healthcare industry. So many people, everybody is involved and everybody has to play their part. And one of the important and critical issues is the time. So whenever we talk about handling it, controlling it, this COVID-19, it is about most precious resource was the time. So act fast. If you lose time, you lose people. We have experienced such thing in diagnosis itself. We were involved only in one part, the diagnosis. So in diagnosis also, the time is so critical. If you lose some time, then you see there is a series chain of reaction and then you can't control it. So preparedness is the key to save time and save people. This is the first lesson and the most important lesson that SARS-CoV-2 virus has given us. And I will share my experiences, whatever we were involved, so that now preparedness actually, as the topic also says, the preparedness for pandemic. Preparedness is a very, very big word for a pandemic, actually. Everything we have to do is the preparedness for pandemic. Now, WHO, they have number of documents and the plans for different nations according to their economic status and social status. They have developed plans for basic plan for controlling the pandemic. So you have to have a number of issues they have highlighted, and we have to adopt it. And actually, medical people like medical institutions like National Institute of Virology and NCDC, they are all involved in planning all those things. But general public and other healthcare and veterinarians, they are not involved so much into this. But now it is everybody's problem. So we have to know what is a preparedness plan and how we can prepare. And as we talk about the preparedness for pandemic, it has to be multi-sectoral involvement is there. And some critical issues are there like antiviral drugs and vaccines. The countries, they stockpile the vaccines and drugs also, antiviral drugs also. And critical supplies like ventilators, N95. For this, we have so much of experience now in these 4 months. Initially, when we started on 12th April with this diagnosis, so usually because we are a BSL-3 lab, so we have this N95 mask, and because of our experience in the H5N1 highly pathogenic avian influenza outbreaks, so we know that suddenly if there is an outbreak, you don't have time to buy new things. You should have it already. So you have to give diagnosis within 24 hours or 48 hours like that. So samples will come flying from by flight, and then next day within 24 hours. So at that time, you don't have time to go to the market or ask some supplier to give us N95 mask or anything like RNA extraction kit and everything. So similar thing actually happened. So we had something, but we were not prepared for an epidemic, an outbreak of such a large size. So we were still in numbers, actually, we were still feeling so short actually. So we had some supplies. So we utilized it. Then we came to know that this RNA extraction kit and the real-time diagnosis kit, everything becomes so scarce. And when you are losing time, then you are increasing the disease also. So these things are very much important actually. And on a regular basis, you don't buy such things, but we have a plan in our lab that for any outbreak, anything, we have to have some stockpiling of these, the diagnostic kits, the RNA extraction kits, and other things, N95, any other biosafety supplies also, like PPE kits are there. So they have to be there in the store, so that you can start quickly and don't lose time. And research is another aspect actually, and during our research programs, planning our research programs in last 20 years, since year 2000, actually we were again and again asked why you are working on a disease which is not present. So it is not a problem, it is not a farmer's problem, why you should work actually. And we had to answer, policymakers actually, they will ask, it is their duty to ask. Say, if the money is going somewhere, then they will ask, okay, why you are spending so much of money on some kind of preparedness of some disease or some problem which has not happened. So this is not the case. It should not go like this. So we had to actually impress upon that if something happens, then we will not have time to react and then respond and even plan also. So it has to be done something beforehand, something happens. So this is the way pandemic preparedness is. You have to have vaccine candidates, you have to have the diagnostics prepared, and you should be going for surveillance and monitoring also, like surveillance of bats for Nipah virus, SARS virus. We should have been doing it actually in a regular basis, because these are some of the viruses, they can anytime they can jump and then to human beings and cause outbreak. They will not give any alarm actually. As we know, recently in Kerala, there was a Nipah outbreak. There was, so suddenly if something is there, you don't find time to respond. If you are not prepared, if you don't have a diagnostic in your lab, you will not start designing the primers and ordering them. You should have there in your hand, so that next day actually you can give diagnosis, and then only you can control the disease. Otherwise, it is not possible in the regular manner, that the problem comes and then you plan it. So now the other concept is the One Health concept, which every veterinarian should keep in mind. What is our role in this One Health? So we are not, as veterinarians, we are not there only for animal husbandry. We are also for the human health. So as we know, at the juncture of human and animal, we have this comparative medicine. Some people must be doing some research work or some kind of work in comparative medicine, and social sciences, they are also involved. Engineering also involved when we talk about the environment. The pure scientists like biology and ecological aspect. The ecology of viruses, they are very, very important now. And those viruses may not be affecting or causing any disease in animals actually, but still viruses, pathogens, we have to study them and then their ecology also, so that we can predict some future diseases. That is why it is important. So we have to tell our policymakers, the funding agencies, when we are putting up the projects, that these are also why these are important to study, so that next day if there is some problem, you have some kind of preparedness. You already know, you have some knowledge about that virus, you have some kind of preparedness, so that you can handle it. Suddenly if a new thing comes, we will not be able to respond to the emergency. So that is why, and as BBSc and AH graduates actually, we should not just shy away from anything if we have to do for human diagnosis or even human treatment if it is required. In such an emergency actually, there are no lines between veterinarians and medicos. Whosoever can do something should be doing it, so that we comprehensively we can, everything is for human actually. We treat animals, we take care of animals ultimately for ourselves only. So there is no divide actually. We should not be seeing any divide between human health and animal health. They are so much interrelated. As food animals, we have to take care of animals, then only we can have healthy human beings. So this is, as a concept and as a role of veterinarian, we have to understand this and conceptualize this, so that when we are asked to do something, then we are clear what is our role here. As an animal, for animal husbandry, we have a different kind of role as for food producing animals. But for zoonotic diseases where the human health is involved, you have a different outlook. You have to have a different outlook. You don't see, like, so many times we were asked, why are you going for this surveillance of the bats? Bats are wild animals, so many, like crows. Crows have been found infected with H5N1 viruses, and our lab has diagnosed it. So we are there for poultry and the food birds, the poultry for commercial purposes and backyard birds, but not for crow. But still, we have to do these things because it is not about what is the area of your work actually. We have to take care of the society and human health also. Wherever there is a risk of human health through animals, we have to play our role. So that is the concept we have to carry as veterinarians. Then, as we see number of examples between wildlife and human beings, there is so much of crossing of the viruses like SARS, Ebola, hantaviruses, monkey, Lyme disease is there. Then we have few viruses which can be transmitting between wildlife to livestock also, domesticated animals also, and then human beings also. So we have in this list rabies virus, West Nile virus, tuberculosis, anthrax, tularemia, plague, even influenza is also one of them, which is quite important actually. We'll talk about it. And then we have this BSE, E. coli, cowpox, Rift Valley fever. Rift Valley fever we have not seen much in India actually, but this is also one of the potential zoonotic disease, and it can anytime it can actually create problems. Now, after having this current scenario, and then what could be the characteristics of a future pandemic? Now the scenario keeps on as the socio-economic development takes place, the science also grows. Now the science is at a stage that bacteria is not a problem. Any kind of bacteria, of course, AMR is there, antimicrobial resistance is coming up, but bacteria as a microbe is not much a problem. You have a number of antibiotics, you can control it, you can easily control it. You can't, it cannot get into the size of pandemic as earlier used to, like Yersinia pestis that created, that plague. So viruses, our research has not gone into that level that we can have some antiviral which will be universal for all viruses, or which will cover so many of viruses, or which is so effective that it works like an antibiotic. So that research into the antivirals is very, very important. And about the future pandemic pathogens, a number of articles and book also, these two that are shown in this slide, Pandemic Pathogens and by David Kuhlman, Spillover, the animal infections and the next human pandemic. So a very good read is there. These are available on Amazon also. So as we see the characteristics as microbes, actually, viruses are the most important for causing future pandemic as a pathogen. And if we talk about genome, DNA viruses are not so much risky than the RNA viruses because of their high mutability. RNA viruses can mutate so frequently, but DNA viruses cannot. So because of that property, RNA viruses, most of the pandemics, most likely it would be a RNA virus that is there. Then mode of transmission is very, very important actually, that how it spreads actually. So respiratory route has been identified as the most efficient way of spreading the infection. And this is chiefly because due to the fact that interventions to interrupt this method of spread is very, very difficult. You sneeze, you can't stop the breathing. So if you are breathing actually, so you are transmitting the virus in air, and it may go to other person through contact also or through air also. So airborne infection, it is very, very difficult. You have to that is why the use of PPE and that face mask is why it is so important. It is because of that virus can be transmitted in air by this breathing also. So that is very, very important actually. And timing of transmission also, as we saw in COVID, before the start of symptoms in the patient will start spreading the virus, emission of virus is there. So that is, if the pathogens are shed before the onset of symptoms, it will likely cause the pandemic. Because if you symptoms comes first and the shedding of the virus is later on, then probably because symptoms are there, he will get the medical treatment and this shedding of the virus can be controlled. And at least he will be isolated, the patient will be isolated. Now here in this case, actually, you don't isolate a patient unless you get symptoms. So once the symptoms are there, before that he has already infected so many people. So that is why this timing of transmission is also important. Again, many of the host factors, they are also important, like presence of receptors for the viruses, whether they are ubiquitous in respiratory tract or wherever. So that those factors, they are also going to play what kind of future pandemic can happen. And most likely it is almost sure that it will be a zoonotic in nature. So let us have a look on the viruses with pandemic potential as per the WHO and as per OIE also. So as they enlist, they still find that influenza is one of the most deadly virus which can become, which can lead to a pandemic much greater than this COVID-19 also. So that event of adopting to the human beings, and that has not happened so far, so that human to human transmission is still limited in among influenza viruses, but it is very likely actually. So among H5 and influenza viruses, most of the H5 viruses, H5N1 viruses, which have originated from China and the neighboring countries, they have caused most of the infections in the Middle East. And human infections are often associated with severe pneumonia and mortality greater than 50% also. So once they adopt to such a chain of transmission from human to human, then the mortality will be much, much more actually. So at that time, the hospitals and the medical care system, it is so fast and unpreventable that the hospitals and the medical care system, they can't cope with that speed, and the preparations has to be there. So that is why since last so many years, WHO and many other developed countries in India also, they have always a preparedness plan for influenza. So they stockpile some vaccines also, and some vaccine strains actually, not vaccines, but antivirals also. They should always have a supply of Tamiflu, that is antiviral for H5 avian influenza, and that is a must, because anytime it comes, so you don't have time to then plan and then buy things. Then comes the avian influenza H7 virus, which we have not seen yet in India. But these are rated actually more risky than the H5 viruses currently, because some of the newer strains of H7N9, which emerged in China in 2016, their pathogenicity was changed, and they continue to be associated with the human infections in China. And the average mortality rate was about 40%. And the currently studied viruses, H7N9 is one of the is considered to be having a greatest pandemic risk based on the historical outbreaks and its characteristics. So that is why these two viruses are still, we have to go first, because as veterinarians we have to take care of poultry, and we are giving health care and we are also controlling influenza viruses. The field veterinarians, they are also involved. The state level diagnostic disease diagnostic labs, they are also involved. They have a mandatory plan for serosurveillance and other things, and they have to send some samples to NIHSAD also regularly. So when in the state where outbreaks of H5 they are there, then the system gets activated, and then they will be doing it because there is pressure from. But we have to be vigilant. We have to be vigilant every time for these viruses. And then some other viruses, viruses of causing hemorrhagic fever, viral hemorrhagic fever like Ebola viruses. They're found in non-human primates like monkeys, gorilla, and chimpanzees. Many of these viruses, they have been recorded in Africa. The outbreaks have been there in Africa and other African countries like Kenya and other countries. But anytime they can come to our mainland, Indian mainland also, and we have to be vigilant. And Lassa virus is one which is transmitted through rodents. Then Rift Valley fever, again, it can affect the cattle, buffalo, and even camels also, and it can be transmitted to human beings from these animals. And the other is the Marburg virus disease, which was found in African green monkeys. And the fruit bats actually, they are reservoir for so many viruses, Nipah virus also, and so many other. SARS virus also remains there. And so they are very interesting subject actually. As far as new study of new viruses are concerned. So we must have a plan for surveillance of fruit bats and other bats, different bats. What kind of viruses they are harboring and how they are changing, how they are transmitting, and what conditions they can jump into domesticated animals or to human beings. This is an interesting research topic and very, very important thing. Then we have CCHF. CCHF, we had outbreaks in Gujarat, and in our lab we got samples of ticks and serum from cattle and buffalo also to look for in serum antibodies, and in ticks the virus, whether CCHF virus is there or not. So whatever samples we got, we didn't get anything as such, but we have to have a comprehensive surveillance plans for these diseases. At the face of it, actually, it is not a problem of animal husbandry. They are not affecting the animals. But for human beings and for prevention of these emergence of new viruses, we have to have a surveillance plan. So veterinarians, they can play a very much important role. As a researcher or in institutions and universities, you may have a project or research problem for looking for all these things. But as a field veterinarian, they must be looking for anything, any symptom or any disease which they doubt they have not seen, and must doubt and send the samples to the reference labs wherever possible, so that we can catch these emerging viruses from their origin itself. Once they are allowed and a sizable population of the animals is already infected, then it will be too late actually. So that is why I said that field veterinarians, they have their own role to play in this prevention of pandemic and prevention of emergence of new viruses. Diagnosticians, laboratory workers and laboratory diagnosticians, they have a different role. And as researchers, we have our role to play to study these viruses, or at least have diagnostic preparedness ready, so that if we are asked, we should not take much time in responding. So that is how. Then after this came the coronaviruses. Coronaviruses, they were known to be having the pandemic potential, and then MERS-CoV virus was one of our enlisted virus in our lab, so that we had this preparedness earlier, not exactly for the SARS-CoV-1 and 2, But for MERS, we had this preparedness that coronaviruses can cause such problems. So this is the current scenario of 14th August, and the whole world, most of the countries, they are having the cases, and many are in deep red. That is more than 30 cases per 1,000. That is too much high, actually. This is and whether this is the peak or still we have to see the peak, we don't know, and we have much to do. But before that, whatever is being now, everybody's focus is on COVID and SARS-CoV-2. So a lot is being done and will be done in SARS. So many research projects have come. So many vaccine things are coming up. Maybe one day we will have the vaccine. All this, but that should not. That should not lead us to just focus on this SARS-CoV-2. There are so many other viruses, especially the influenza viruses, that have to be taken care of. Animal influenza viruses that is to be taken care of, so that we don't allow them to become a pandemic virus. So that is our role, actually. Then just sharing our experiences, that how we got involved in the diagnosis as veterinarians in veterinary labs. So many veterinary labs across the India, they started diagnosis of this SARS-CoV-2 for human samples. So Nagpur Veterinary College is one under MAFSU. Then we have DUVASU, Ludhiana. Then we have INRDDL, which is the Regional Disease Diagnostic Laboratory for animal diseases, actually, in Jalandhar. They are also involved. These laboratories, they are listed in the ICMR website that they are doing the COVID diagnosis. So then Mathura Veterinary College, they have also been involved, and I think they are regularly doing it. From ICR, we have in Nagar campus, IVRI Mukteshwar also, they are doing this daily, they are doing the testing. ICFMD at Bhubaneswar and NIHSAD, our lab also at Bhopal. We started in 12th April. So earlier there were some issues, actually. We were also confused whether to take up this assignment and not so much. We had a meeting with the medical people, and the legal issues were also there. So we said we can do the testing, but we are not going to sign a report of a human sample because we are not authorized to do so. I don't know what is the answer till now, but I have been sending the reports as others have been doing to some 80 to 100 samples per day. We are sending the report of COVID-19 and to the IDSP and the in charges there. And sometimes some individual patients, they also ask for that, can we get a signed report? So from ICMR portal, you can generate a report and sign it and give it to them. But that remains a question whether as a veterinarian we can sign the diagnostic report. But when it comes to such a big calamity and such a big huge problem in which everybody is involved, we can't just shy away that we are veterinarians and we will not do this thing and that thing. Whatever we know, actually, we have to contribute. So at NIHSAD, how we planned that for SARS-CoV-2 viruses, we had different teams. Actually, this was one activity in our lab that in which all the scientists, they were involved, right from senior most principal scientists to the S1 scientists also, all were involved. All are involved currently also from the start. So sample receiving, coordination and reporting was one team. Sample processing is another team. They will process the samples, and RNA extraction is a different team. They will do their job. That is how we are able to manage these activities, that for 2 to 3 hours, every team will work and then they can take rest. So that work is not stopped. Nobody is exhausted. So on daily basis, it is sustainable. So daily basis, you have to work for 3 to 4 hours, one team, and then they can do their own business and take rest. So real-time PCR is the most critical part actually when you have to give the diagnosis. It is a very sensitive test, and still we had seen problems of supplies of the kit also, and so many. I mean, we have changed so much. We started with a local company's kit, and then we got NIV's kit also. Sometimes we got imported kits also, like from LabGun. So right now we have seen that for any of the good kits, especially for the RNA extraction also, we used to use only QIAGEN kits for RNA extraction, especially for diagnosis of very critical nature of this type, that we should not use other kits. But we saw that we never got QIAGEN kit after the COVID-19 because the supply is so short. These companies, they can't make it in such a huge volume, and because they are, that is why the PM's call for Atmanirbhar Bharat is very, very relevant here also. We finally saw that we came down to RNA extraction kit provided by the Indian companies only. Hymedia, this RNA extraction kit we are using, and another company's, this Indian company's real-time PCR kit we are using. The primers, probes, they are provided, manufactured by them. So this happens when the emergency comes, and then in the world everyone is affected. Every country will first satisfy and they fulfill their own requirement first, then to export the kits and these supplies. So likewise for N95 mask also, we were using the 3M mask, always purchasing that one only, that this is the best quality and whatever. But after COVID-19, we never got that kind of N95. They were in short supply, and then somehow we had to have this local made, and whatever good options were available, we have to buy that, and they also work. So here, actually, this concept of Atmanirbhar given by the government and PM is very, very relevant and important, that for these, when war comes or some warlike situation or the pandemic comes, then you have to depend, you can depend on your own resources. You can't look, nobody is going to supply. They will first have their own things fulfilled, then only they can give it to you. So that is the case. So we should have facilities for making our own instruments. We still, in research, actually, we still have a fancy for imported things. That should go away. Make in India should go ahead with that concept. Then looking at the numbers, we have so far tested some 11,000, nearly 12,000 samples, human samples, from which 865 were positives. And we have done some whole genome sequencing also from 9 samples using nanopore platform that was recently optimized by our scientists. And then what we do at ICAR-NIHSAD, this is regarding, I mean, how we carry this concept of preparedness. Our main vision and the mission and the mandate is talking about the preparedness for some diseases which are unknown or which may come in future. That is the main thing actually. So we have been living this thing right from beginning. And like I have before me some very good examples, like for avian influenza. Once that outbreak happened, initially the diagnosticians, they were handling that. They denied, no, it cannot happen in India. No, it may be some NDV virus. But when the samples came within 24 hours, we diagnosed. How we diagnosed it? How our scientists, they diagnosed it? Because 2-3 years back itself, we had the primers, probes, and everything prepared for avian influenza. At that time in 2002, there was no talk of avian influenza. Many of us didn't know that this is a disease like this, and this can affect the poultry in such a devastating manner. But still, we had everything prepared. Then only we could respond. So similar way, for future things, we have to get prepared. Then what we do at NIHSAD, our activities as preparedness, you just see the diagnostic preparedness. So we have this OI recommended test, and for every disease, whatever is there. So we get the primers for the PCR, at least that much is there, or ELISA kits if they are available for sero surveillance or for antigen detection. That also we keep and actually in our stock. And then another kind of preparedness is the preparedness with the vaccine candidates. So you may not need we have never utilized or used avian influenza vaccine, and we have never recommended also. But still we made some vaccine candidates for avian influenza, because sometimes if the spread is so huge and widespread of outbreaks are there, then at least we should have the candidates ready if government asks, okay, you have to provide the vaccine. So at that time, if we start developing the vaccine candidates, it will be very difficult, and the time will be gone, then all poultry will be finished. So this is the concept. Then surveillance and monitoring is also part of the preparedness. When you just keep on monitoring for new diseases using the accepted diagnostic tools, or maybe through the metagenomics approach. Then we also started developing our own diagnostic kits as per the needs by the state DI labs or wherever. So recently we have developed some diagnostic kits also as per the need. And these are the list of diseases, emerging diseases and exotic diseases for which we have diagnostic preparedness. Some of these diseases have never been reported here. And recently African swine fever was there. So this is also a very typical example. It was supposed before this was in March, actually March 2020, that the suspicion of this disease was there in northeast states. So when the samples came, we had everything ready, and within 72 hours we gave the diagnosis, and the sequencing was also done to confirm the first outbreak of African swine fever in India. So that is the kind of preparedness we need for at every level, not at NIHSAD only, but at every level we have to have this kind of preparedness, so that we don't wait that a disease will come, then we will study about it, and then we will prepare something. Everything should be prepared beforehand. Then only we can handle it, and then only we can check the losses and prevent the losses of farmers and human life also, and economic losses also. So these in bold, these diseases in bold, they are actually of zoonotic nature. CCHF also, West Nile fever, swine influenza, Rift Valley fever, and so these are then more important from that point of pandemic point of view. Then some of the diagnostic kits like indirect ELISA kit for avian influenza antibody detection we have developed. Similarly, PRS came as a problem in northeast states in 2013. Now, within few years, we were able to develop this indirect ELISA kit for serosurveillance. And then we have a multiplex RT-PCR kit for avian influenza A subtyping and differentiation of H5 and N9. And then, interestingly, we also prepared some vaccine for this. For this activity, we had to face so many questions also, because at the same time, when we were saying that we do not recommend vaccination against avian influenza by culling and by quarantine and by biosecurity, only this disease is ideally to be controlled, and we will follow that. But at the same time, we were also developing. So this was the kind of preparedness. We prepared for H5N1, we prepared a DIVA marker vaccine using the reverse genetics technique. We H5N2, and then for H7 also. H7 another kind of, because H5N1 was there, so we could use the genes of the field viruses to develop this H5N2 virus. But we didn't have this H7. And when we asked from other countries, they denied, no, you can't. We can't send you the virus or even genes of the virus. So we synthetically prepared the genes, constructed, and then developed this virus H7N2 non-pathogenic virus as a vaccine candidate also. So we hope that we will never have this chance to use it. But as insurance and as preparedness, we have to have some kind of vaccine candidates already available with us. And some few other vaccines under development are for PRS and bovine viral diarrhea, for low path, even influenza H9N2 also. And we are also working on a concept of universal influenza vaccine, because our studies told that exhibited that these vaccines will work only with the clade which is matching with the H5 gene of your vaccine virus. So that is why we started working on that also. So yeah, only 2 slides more. And then this, we had this reverse genetic system purchased from University of Wisconsin, USA, and then 8 plasmid system also from USA, and utilized this for developing the vaccine candidates. Now we are planning for the vectored vaccine also. All these actually, a vaccine development platform should be ready with the labs actually and the research institutions, so that whenever the requirement, such requirement comes, they can easily and quickly respond to it. So that is the strategy to handle disease outbreaks and control them, and then provide the vaccine development whenever needed. Then this is the site-directed mutagenesis we did for converting highly pathogenic avian influenza into low path. So all these techniques we learned from US, while some of us, we went to US for training in normal Borlaug fellowship, and then we applied it here to have our preparedness on influenza vaccines. This is also the genes amplified, and then transfection in 293 T cells so that we can rescue the virus. We can rescue the non-pathogenic virus, which can be used as a vaccine candidate. And then to conclude further, that as veterinarians, we have to see our role in different scenarios and different places. We already know it, the control of food-borne pathogens. Food-borne pathogens are very, very important. Vector-borne diseases, like many diseases, are transmitted through vector. So CCHF is one example. Hygiene and sanitation in slaughterhouses and live bird markets are very, very important. And most of the influenza transmission and recombination events, they occur during the live bird markets actually, because from you have poultry from number of sources, they come, and different kind of viruses they can combine there, they can be transmitted. So these things are important. Then use of misuse of, we should avoid the misuse of antimicrobials in production of animals that actually that poses human health hazard. And then surveillance for emerging zoonosis, this is very important, and it's important from field veterinarian to laboratory diagnosticians, that we should be looking for new things actually. So anytime anything new will struck, and you will believe, okay, no, why it should have, why I should think that a new thing is coming up. I have never seen such a thing. You must doubt every time that if something new is there, let us check it with the reference lab, or do whatever resources you have, so that, because these things are happening, and God knows when we are faced with these new viruses. So then awareness among animal owners also important. They don't know about CCHF. They may not be knowing this is a new disease. So for new diseases, they must be knowing that what is the risk when they are handling the animals. Even domesticated animals, ticks are there, whether they can have infection from them. Then we should also participate in One Health activities at the animal and human interface as veterinarians, and with the medical people wherever possible, in whatever role. Then in case of emergency like pandemic of this COVID-19, we can adopt any role in the medical field also, as we have some of our veterinary friends, they have shown us that they are doing diagnosis and then giving the result for human pathogen. So that is the gist of it. I think I have taken much time actually, and I hope some time is left for some discussion also. I want to hear from everyone. Thank you so much.
Thank you, sir.
Yeah.
First of all, I would like to thank you for updating us on the role of veterinarian in this pandemic. I also appreciate that you highlighted the role of veterinarian in this current pandemic, and now I can realize that how busy you people are being a veterinarian to deal with current pandemic. We are really very lucky that you find some time and come on this ISVM forum to, it means, update us about the role of veterinarian, how to tackle the pandemic. Sir, one important thing I would like to share today. For today's webinar, senior life member of ISVM, they have joined. Among them, Dr. Sushuban Roy sir is on forum. Dr. Abdul Samad sir, he is also on forum. Dr. Umesh Dimri sir, he is also on forum. Dr. D. Mangal sir, he is also in forum. Also, sir, from your institute, Dr. D.D. Kulkarni, he also joined.
Yeah.
Yeah, I also appreciate the active participation of EC member of Indian Society for Veterinary Medicine. That Dr. Ravi Jadhav sir is there, Dr. Dheeraj Gupta sir is there. And I also appreciate the participation of young energetic ISVM life members like Dr. Gautam Bojner sir, Dr. Pazai sir, Dr. Suresh Patel sir, Dr. Akhilesh, Dr. Poonam Vishwakarma, Dr. Pankaj Kumar. All they are joining sir today for your webinar, sir. In this time, sir, I would like to take some questions. They have been dropped by the participant in the drop box. Okay, sir. First question is that from Dr. Manish Kumar. He is willing to ask, is there any report on antiviral effect of herbs like neem extract against COVID-19?
Yeah, we had some work done by Dr. Rita of neem extract and tulsi extract on influenza viruses. So that is there, but on COVID-19 we have not worked, but somewhere in people are working actually, and these actually we have seen that these extracts like neem extract, and their action is actually not so specific to the virus. So if they are working on enveloped viruses or RNA viruses, then it will work for a number of viruses like that. So it is not something very specific. So I don't know whether somebody has tried or not, but I know that many of the studies they are going on. Only thing is that for working on COVID-19 itself, there is so much of restriction because it is a pandemic virus. So people who have access to this virus, either they are basically the laboratories which are busy in diagnosis. So let us take our case also. We had number of proposals for working on this virus, but only for few we could respond because we are so busy. We have the opportunity, we have this thing, but we can't transfer the virus to some other lab if they don't have the biosafety facilities. So that is the case. So these studies, they are restricted by the requirement of a biosafety lab for working on these viruses. But there is so much potential in these herbal antivirals, and they can do a good job, I think. And one must, I think, in future we should have something like that.
Okay, thank you, sir. Sir, again, there is a question from Dr. D. Mandal from ICAR.
Yeah.
He is willing to ask, if coronavirus vaccine is developed, if it inoculated into human patient, how many days it will protect from infection?
Yeah, that is difficult to, you see, as we talked about the vaccine platforms, the different vaccine platforms are there. Now for coronavirus, as you know, that mRNA vaccine is in the front actually, that is coming up. Then you have inactivated vaccine, and then you have subunit vaccines also. So it depends on the the kind of platform which you are using. If it is a mRNA vaccine, we have no experience about the mRNA. The world has no experience actually. This will be the first mRNA vaccine, I think, which will be used practically in human beings. And then most of our experiences have with the killed vaccine, the simple inactivated virus vaccine, which was made by this in India. It was made by Bharat Biotech, and they are still doing this trials, and it will take, I think, 6 months or 1 year to complete the trials, and that is the case. So I don't think more than 6 months it can in generally we know that any inactivated vaccine you don't have, because you are not propagating that virus. So it is, everything is killed. So it is a protein, it is a inactivated antigen. So maximum it shows in case of influenza, also in other viruses also, a killed vaccine gives protection up to 6 months to 1 year. Most 1 year is also too much actually. Most appropriate or optimal protection is given up to 6 months only. After that it weans off and then reduces to up to 1 year. So that is the case. So until unless you have a live virus vaccine, like in NDV case or other vaccine, where a live attenuated virus is there, so the problem is there the scenario is different. The virus can replicate, it will not do harm, but it will keep on stimulating the immune system, so that your immune status is maintained for a longer time, like that. So all the live virus vaccine, they give a better and longer protection than the killed vaccine. Now, because of the nature of the virus and its risk, biorisk also, we can't think of a live virus vaccine for such viruses, and especially for this for influenza also, for high path influenza also, we don't recommend any live virus vaccine, only killed vaccine. So with our experience with killed vaccine is there, and from world over, they say that it will allow the immunity up to 6 months only, and it takes at least 15 days to so that sufficient antibodies they come, so that it can protect. So it depends on so many things. It will be difficult to say right now. Only the the research teams which are involved in the vaccine trials, they can really specifically answer this question.
Thank you, sir. Thank you. Sir, again, there is an interesting question from Dr. Srishti Singh. Question is, there have been several outbreaks of zoonotic diseases in the past decade, but none have gone on to become a pandemic. What is different this time? So COVID spread so fast and quickly.
Yes, that is an interesting question, and we had some highlight in one slide. What are the characteristics of a pandemic virus? So world over organizations, they had this exercise also. So what is the characteristics? It has to be a respiratory virus, first thing. If it is transmitted through insects or through other vectors, vector-borne diseases, they cannot spread so fast, because the vectors can't transmit the virus from one animal to another, human being to human being, or any species to species so fast as the virus, which can be spread through breathing only, or through air only, or through sneezing, like that. So that is one of the characteristics. It has to be a respiratory virus once. And then the receptor, the host factors in this COVID-19, actually the receptor, that host factor was very, very important. You see, its attachment, its affinity to the our human receptors, which are so much ubiquitous in the respiratory system also, and some vital organs also. So much of those receptors are there that wherever, when it enters the body, it can attack every vital organ it is attacking. So that is the case. So that host factor has become so much important in this COVID-19. That is why it is so deadly. And then for transmission, for spread, because it is a respiratory virus, so it can spread easily. And the amount of virus that is generated also is also important. So when you have a viral infection, then every virus behaves in a different manner, and the amount of virus which is shed, and how much is the infectious, how minimum number of viruses can affect the other person, that kind of mathematics is very, very important here. So once that is at a threshold level, beyond the threshold level, you can't control it. And then symptoms, as I talked about, that before the symptoms come, if the virus shedding is there, then again you don't see that actually. So you don't isolate the patient. You can't quarantine it. So before a patient is identified by test or by symptoms, it has already spread actually. So the damage has already been done. So that is why it is difficult to catch it and then control it. So that is why it takes the shape of pandemic. It spreads so fast. Even if government is doing, they are hell bent on controlling that virus. They are doing every kind of job, like from lockdown to banning all the travel, etc. Still, it is there. So these characteristics actually they make the zoonotic viruses. Every zoonotic virus may not have that pandemic potential. That is for sure.
Thank you, sir.
Thank you.
Sir, again, there is one interesting question from Dr. Rajkumari Mandakari Devi. She is a microbiologist in Northeast region, sir. She is willing to ask, after the 2003 ARS outbreak, it is well known that coronavirus has high pandemic potential owing to its high adaptability and can cross the species barrier. Still, we fail to control it. Why?
Yeah, that is the question I am also putting to because we ourselves at this institute we were also facing, because importance of these things and these pandemic viruses and their preparedness was never realized actually by the policymakers. So whenever we start some work or something that we should do like this, we were often asked, what is the need of this? This is not the problem. We were focusing only on the current. We are adopted actually and accustomed to focus on the current problem. What is the problem today? Let us focus. Now we are focusing on COVID. Everybody is focusing on COVID-19. So they don't know what is going to come in next 5 years or 10 years. So if we focus, if we plan ahead, then only we can catch it. So that is the problem. But after, I hope, because after this experience of COVID-19, even the policymakers and the government, the government itself of every country, they realize that the importance of having the preparedness for these viruses.
Thank you, sir.
Thank you.
There is one question from Kumar. Sir, will the vaccine developed be effective for COVID-19 if RNA virus can mutated?
Yeah, that is, it depends actually that what is the protective antigen. In case of SARS-CoV-2, it is supposed that the spike protein, the antibodies against the spike protein, they should be protective actually. That is important antigen. And in that antigen, if the gene of that antigen, it should be more stable. If it is stable, then you have a lesser problem. Like unlike in case of influenza, in influenza, HA gene is highly mutable, and it is the protective antigen. That is why it is very difficult to prepare a vaccine against H5. So you make one vaccine today, another strain or another with some changes in only HA protein will come, and that will work only 50%. That is why. So it depends because we don't know much about this virus, still studies are going on. When we have some vaccine trials with of SARS-CoV-2, then only it can be definitely said that which is the protein which is more protective and stable also. If it is not stable, if it is highly mutable like HA of the influenza, then it will be very difficult. Again and again, for influenza, you know that for vaccine preparedness, WHO has a plan. What they do that every year they change for seasonal influenza, not for even influenza. For seasonal influenza, because influenza is a RNA virus and HA is the protective antigen, and that is highly mutable also. So every year they change the strain. So they update the strain. Now this year this strain will work. So you will have a problem like this. So most of the RNA viruses, they pose such problem, but it may not be exactly same as in case of influenza. If it is less mutable or more stable, especially the protective antigen, then it will be a lesser problem.
Thank you, sir.
Thank you.
Again, sir, there is one question from Dr. Vijay Lakshmi.
Yeah.
She has two questions in Dropbox.
Yeah.
First, what is the consistency of different kits being utilized for COVID diagnosis, and which kit is most efficient? And
For
second question, I'm repeating question again, sir.
Yeah.
What is the consistency of different kits being utilized for COVID diagnosis?
Yeah.
Which kit is most efficient? This is first question. The second, she is willing to ask, coronavirus vaccine is available for dogs. Will it give cross protection as far as the human beings are concerned for to COVID-19 for humans? Or any research has been planned in this area?
Yeah, I can answer about the first question actually. Actually, for vaccine, there is not much known. Still, studies are going on. No vaccine study is complete. And we ourselves not initiated any vaccine studies also. But for first, I'm sure that she asked that which kit is, a number of kits are available. But in our experience, we saw that the reagents provided by National Institute of Virology, Pune, they are the best. They will never, they give so much clear-cut results. So we always, whatever kits we are using, commercial kits we are using as per the availability, because these are supplied by the ICMR Institute. So we don't buy directly. We have no, system. So they supply either the state government or the ICMR Institute. They will supply these kits for RNA extraction also and for real-time PCR also. So whatever they have this kind of supply, we will have to take and use those kits only. So in our experience, we have used number of kits only. NIV kit is most reliable, but other kits are also good enough. And sometimes if there is some doubt, we always keep this NIV kit. So, but the doubtful samples will be easily recognized and identified experienced person who is experienced in, real-time PCR. So you don't have a typical curve of a positive, only one gene is coming positive like that. If some doubt is created, we retest these samples with the NIV kit, and that gives a very unequivocal result. So that is how we deal with this thing. So we recommend, as somebody would ask, then NIV kit is the best still.
Thank you, sir. Sir, there are again almost 5 to 6 questions, but I will take 2 questions, sir.
Yeah.
Yeah, there is one question from Dr. Siddharth Shanker Pathak. He is willing to ask, what is your opinion on vaccine developed by Russia?
Yeah, Russia actually, that is for all vaccines, I would say. We can't give any opinion because no experience with the COVID vaccine. And the studies are yet to be. We have to see the effect, and then we have to form an opinion. So to form an opinion about a vaccine, we have to see the results. We are not seeing the results actually. Whatever is given by Russia or whatever is coming in the newspaper, on the news or any article, we can, because when such a big problem happens, then countries are there, they are political also, and then political issues and countries' own interests, they also came. What is true, what is not true, we can't say. Sometimes it seems that if some country is announcing, okay, this is a vaccine made, developed country like US or anybody, they may not feel good. They may say, oh, this is we learned a few comments from this US also, that this is going to be useless or something. But until unless we see its use in field means in human beings, and for a long time, at least for 2-3 months or so, then only we can say something about it. Otherwise it is speculation only. So everybody can do speculation, but confirmately, so we can say something only after we have seen the effect.
Thank you, sir. Two again interesting questions are there, sir.
Yeah.
One is from Dr. Manjura madam, and one is from Dr. Rotvik. Madam is willing to ask how long will be actual incubation of COVID-19, and second question is also on the same line. Why asymptomatic patients are there, or in patients who are getting, as far as the COVID, asymptomatic patients they are reported?
Yeah,
that is
yeah, that incubation is known actually. 9 to 10 days are there, but it varies also. So nothing is certain, but most optimal and this is incubation is 9 to 10 days, and within 9 to 10 days one starts developing the symptoms. Some it depends on the immune status and general health status and other comorbidities, the other diseases a person is carrying also. So how easily and how quickly he will succumb to the virus replication there. And then the second one was what was the second question about?
So why some patients they are asymptomatic in nature?
Yeah, asymptomatic. Yeah. It can. So happens that the virus first infects the respiratory tract. So before that respiratory tract is infected and the virus production starts, the symptoms of respiratory illness, they are more like, because we have so many other, the respiratory viruses are very common, like so many respiratory viruses are there. Influenza is one of them. So for so many reasons you have a sore throat or some kind of sneezing is there. So nobody takes that seriously because it happens, it regularly happens to everybody. So that is the case actually, because you don't notice that, because that is so many other viruses are there. So you don't doubt that it may be a SARS-CoV-2 virus. So that is why those are neglected, and that time actually a lot of transmission happens in the initial stage. It is only the when the severe symptoms come. Severe symptoms will come only when the vital organs they are affected, and the spread of the virus throughout the body is widespread in the liver, lungs, and in the heart also. In the circulatory system also it is there. So that is why at that time the severe symptoms are there. Before that nobody is so vigilant, and then nobody will actually doubt about that that this is going to be. And sometimes it is false alarm also. So many patients we have seen that I'm having this fever last night. Okay, how much time I will get the results? And they are so panicky actually, and then it comes negative. So it happens because other viruses are also there in the environment. They are also playing their role. So it is difficult to differentiate in the initial state whether it is an influenza-like symptom or it is SARI. SARI is actually severe acute respiratory illness. So ILI and SARI are different. So most often people, they will have this ILI, this influenza-like illness. So influenza-like illness is very mild and not taken seriously. And because so many other viruses, they cause ILI, so you have no choice actually. So until unless you test it. So that is why testing is becoming so important, because on the basis of symptoms, initially you can't differentiate anything. So you have to test it, then only test will tell whether you are infected with the SARS-CoV-2 or not. But if it is SARI, that is severe acute respiratory illness, then more than 50% chance is there that it will be SARS-CoV-2, like that.
Sir, thank you. Neelesh, I hope you will have. Hello. Is it
Yes,
okay now? इको बहुत आ रहा है मेरा.
Sir. Oh, echo.
ठीक है. Sandeep sir, first of all, I would like to congratulate you because more than 100 comments are available here in chat box. All they are congratulating you. Few of important comments I am just reading for you. Some
Yeah.
people that is very nice presentation, topic presented very nicely, information in the webinar is very good, and the information available in the webinar is very knowledgeable. This information is very knowledgeable. So I would like to congratulate you and thank
Thank
you
you.
for giving us this valuable information, being a veterinary clinical person, sir, or better being a clinician, sir. Sir, again, there are some questions. I will collect these questions and submit to you.
Yeah, sure.
And
I will
after
answer.
getting clearance from you, I will post this question, answers of these questions on our ISPM, whatever the Facebook group is there, on that Facebook group.
Yeah,
So,
sure.
yeah,
Welcome.
sir. I'm really thankful to you, sir. And at the now I would like to request Dr. Gautam Bhojne, sir, to propose vote of thanks.
Thank you so much.
Yes, sir, please. Yeah.
Sir, I'm audible.
Yeah.
Namaste to all. I know duty is more urgent than that of the returning time. Help in webinar success. Gratitude to Indian Society for Veterinary Medicine, along with the knowledge partner, Zydus Animal Health. A million thanks to our distinguished orator, the phenomenal Dr. Sandeep Bhatia, Principal Scientist, ICAR, National Institute of High Security Animal Disease, which is situated at Bhopal. On behalf of the organizer, I take this opportunity to wholeheartedly thank and congratulate him on a wonderful and informative presentation today. The way he explained the topic is outstanding. Our best wishes for all your future endeavors, sir. I must say that we are all inspired by your enormous work, sir. Thank you once again. I wish to express my gratitude to the respectful Dr. D.S. Nauriyal, President, Indian Society for Veterinary Medicine, for providing encouragement and support and for organizing the series of events of webinars. Also, to Dr. R. Ramprabhu, General Secretary, Indian Society for Veterinary Medicine, for moral support and guidance. I extend my sincere thanks to webinar moderators as well as the event coordinators, such as Dr. T.N. Galdhar, Dr. Neelesh Sindhu, Dr. Sujit Kumar, Dr. Shanker Singh, for smooth conduct of this webinar. The words fall short to owe a great deal for technical support of key governance team of DUVAS, Dr. Neelesh Sindhu, Dr. Vijay, Dr. Sumit, Dr. Sumit Mahajan, and Dr. Kumar for tireless contribution they have made, and I admit that the webinar could not have been possible in such a short time without their help. I want to share my views with you that an event like this cannot happen overnight. The wheels start rolling weeks ago. It requires planning and a board's eye for nothing. We have been fortunate enough to be backed by a team of very motivated president and secretary and dedicated ISVM members. My deep sense of appreciation and thanks to all the dear participants who choose to be live with us and attended this webinar with a great enthusiasm. I am also thankful to all our senior faculty today who have joined for this webinar. I thank one and all for being a part of this knowledge sharing quest. Thank you for sparing your valuable time. Looking forward for more such meets. Stay tuned. Have a great evening. Namaste.
Thank you. Thank you,
Namaskar.
sir.
Thank you, sir. Thank you.
Thank you.
There is
Thank
one
you, everyone.
announcement for all the live member of Indian Society for Veterinary Medicine and participants. Every Saturday we are scheduling our ISVM webinar. Next Saturday also ISVM webinar is scheduled. The information regarding the webinar details has been shared on ISVM group and on ISVM Facebook page. All of you are requested to register for the next webinar. Thank you. Thank you, Sandeep sir. Thank you and
Thank
good night
you.
to all.
Welcome.
Good night.
Thank you. Thank you. Welcome.
Thank you.
Thank you so
much, sir.
Thank you.
Thank you.
Thank you, sir.
sir. Webinar was really nice. Thank you.
Thank
so
you.
much.
Thank you.